Hi students, coming to the next topic, bacteria, especially eubacteria, reproduction process. In the last class, we discussed about some of the characteristic features of eubacteria and different types of eubacteria, right? In that case, we discussed autotrophic bacteria and heterotrophic bacteria. The most abundant bacteria are heterotrophic bacteria, and which are very useful to the human beings. Different types of products are produced, right? So that is about that the major criteria regarding the heterotrophic bacteria. And remember the next one, there is a chemoautotrophic bacteria are there. Chemoautotrophic bacteria are main crucial role for the recycling of nutrients. In the last class, I said one of the cycle, right? See, majorly the nutrient cycling process done by chemoautotrophic bacteria, no doubt about it, right? See, once the organisms are died, animals are died, plants are died, okay, once the organisms are died, after that, the crucial role is the saprophytes. Remember, saprophytes are different from chemoautotrophs. Chemoautotrophs are majorly involved in the recycling of nutrients like nitrogen cycle, phosphorus cycle, sulfur cycle is there, iron cycle is there, right? All these recycling processes done by the chemoautotrophic bacteria. And see, the saprophytes also important role. Once the organisms are died, after the death of these organisms, okay, again the nutrients are added to the soil. That function is carried out by the saprophytic bacteria. Saprophytes are heterotrophic bacteria, right? And coming to the next topic is the bacteria and the reproduction process. We know the very first one is the fragmentation process. I said so many number of times. I'm expecting. Suppose when we are working that time, any bacteria is present on the soil. Suppose this is one of the bacteria, right? If we apply the mechanical pressure on the bacteria, or is there any accidental pressure on the bacteria? Accidental pressure on the bacteria can be broken down into the number of fragments, and each fragment have the capacity to produce the new bacteria. Each fragment have the capacity to produce the new bacteria. So that case is called as the fragmentation process. And coming to the next type of reproduction process. So what is reproduction? Production of offsprings. That is nothing but the reproduction process. See, we know there is in vegetative reproduction is there, sexual reproduction is there. Remember, these are the lower organisms. Bacteria are the primitive organisms. Majorly exhibit the asexual reproduction process only. Right? See here, there is a fragmentation process. This is one of the asexual reproduction process, and next one is the endospores formation. See, endospores formation is nothing but one of the bacteria is there. This is one of the bacteria. During unfavorable conditions, whatever the bacteria is there, around the bacteria, there is a thick pore formation is takes place. Around the bacteria, there is a thick pore formation is takes place. Once the thick pore formation is takes place, it have the resistance to high temperature. The bacteria have the resistance to high temperature. See, these pores can resist up to 100 degrees centigrade. These pores, the endospores, okay, or these structures, okay, have the resistance up to the 100 degrees centigrade. So how the bacteria are getting resistance against the temperature? Remember, within that thick coat, whatever that thick coat is there, okay, see, this is a thick coat structure. That thick coat contains the dipicolinic acid. Right? See, the dipicolinic acid is present within that the thick capsule structure, and this substance giving resistance to the unfavorable conditions. This substance giving resistance to the unfavorable conditions to bacteria. Right? So that is about that endospores formation. So how the endospores are produced during unfavorable conditions? There is a thick coat is formed, right? Then this thick coat prevent the entry of high temperature and prevent the entry of harmful rays, right? And inside, whatever the bacteria is there, this is a single bacteria. That bacteria again produces many number of small spores, and these are called as endospores. So these are the endospores, right? When the favorable conditions are kept here, that time, see, whatever the thick capsule structure is there, the thick capsule is broken down and releases the spores. Again, when the favorable condition, that time the thick coat is broken down, the spores are released out, and the spores produces the new bacteria. So the concept is here. Remember, during unfavorable conditions, there is a protection capsid layer is there, a thick coat layer is there. Okay, the thick coat layer form inside, many number of spores are produced. When the favorable conditions are there, that time the coat is broken down and spores are released out, and the spores germination will be takes place, and each spore is equals to the one bacteria. Right? So that is about that endospores formation. Remember, bioterrorism also, the endospores are majority of the bioterrorism also uses the endospores, because the endospores having size smaller than vegetative cells. See, this is the normal vegetative cell size and this is the endospore size. So endospores are having very small size, presence of small size. Okay, the spores are the virulent strains or virulent bacteria are not easily identified. That's the reason why most of the terrorists are using these as the bioweapons. Right? That is about that endospores which are used in bioterrorism. And coming to next is the binary fission. See here we know about this concept binary fission. Bi indicates here 2 and fission is nothing but division process. See, this is one of the bacteria. Whatever that bacteria is there, the bacteria produces some septas-like structures. Whatever the plasma membrane is there, the plasma membrane. Okay, some of the foldings are there. Initially, maybe these are the mesosome structures, but gradually these are developed into the septas because these are separating the cytoplasm of the bacteria. Before the separation of the cytoplasm, for suppose this is the mesosome structure and this is also one of the mesosome structure. Here, what are the genetic material is present within the bacteria? This is the DNA that is attached to the mesosomes, because here enzymes are available, energy also available. That's the reason why the DNA is binded. Then here DNA replication takes place. Once the DNA replication takes place, the one copy of DNA produces the 2 copies. One copy of DNA produces the 2 copies. Then here whatever the plasma membrane folded structures are there, and these folded structures produces continuous septas. Here septas are nothing but separating structures, and these are drawn towards, right? And both these are fused. Then there is a separation of cytoplasm takes place. Once the cytoplasm is separated, later here cell wall separation also takes place. So once the cell walls are separated, once the cytoplasm is separated, once the cell walls are separated, then the one bacteria divided into 2 bacteria. One bacteria is divided into 2 bacteria. So this is nothing but the binary fission. So during the cytoplasm division, before to that the DNA separation takes place. During the cytoplasm division, the ribosomes are separated, the water content and the metal content and storage metal also separated here, right? That is about the binary fission. Remember, in binary fission, DNA replication takes place, DNA separation takes place, septas formation takes place, cytoplasmic division is takes place. Last one is the cell wall formation is takes place, right? That is about the binary fission. So binary fission, in the binary fission process, we can apply here the 2 to the power of n formula. 2 to the power of n formula. See, this is the first bacteria. From this bacteria to this bacteria, okay, 20 minutes are required. For the completion of binary fission within bacteria, 20 minutes are required. Okay, for the 2 cells formation, how much time is required? From one bacteria to produce 2 bacteria, how much time is required? 20 minutes are required. So again, from one bacteria for the production of 4 bacteria, how much time is required? See, this is the 20 minutes. Again here these 2 involved produces 4 bacteria. So here totally 20 minutes plus 20 minutes, 40 minutes are required. Right? So that is about that the 20 minutes time is required for the binary fission process, and 2 to the power of n is the formula. First generation, okay, second generation, third generation, fourth generation, fifth generation. Here n indicates here number of generations. Right? So that is about the binary fission. Right? So this is about that normal vegetative reproduction process within the bacteria. And next is the pseudosexual reproduction process. Pseudosexual means here false sexual reproduction. This one is the false sexual reproduction. Why this is false sexual reproduction? This is not true sexual reproduction. Because any reproduction process, new cells are produced. See, the asexual reproduction means there is no new genes, there is no fusion of male and female gametes. Within asexual reproduction process, there is no fusion of male and female gametes. There is no fusion, right? Within the sexual reproduction process, there is a fusion of male and female gametes. Will be takes place. See here, this is a false sexual reproduction process, because in the false sexual reproduction process there is no new cells production. But genetic material transfer takes place. DNA is transferred from one cell to another cell, but new cells are not produced. I will explain how this is possible, how the pseudosexual reproduction is possible. Majorly 3 steps are the transformation, conjugation and the transfection. These are the 3 methods which are involved in the pseudosexual reproduction process. I will explain now. Coming to the next one is the pseudosexual reproduction process. This is not the true sexual reproduction process, because here there is no production of new organisms. Then why this is called a pseudosexual reproduction process? In all these processes, DNA is transferred from one bacteria to the other bacteria, but there is no new organisms productions. Generally in sexual reproduction process, genetic material is transferred and new organisms are transferred. But in this process, genetic material transfer takes place, but no new organisms productions. Right? See, coming to the very first is the transformation process. Transformation is nothing but very, very easy to understand. See, this is one of the test tube. The test tube contains one of the bacteria. The bacteria is died. This is the bacteria contains genetic material. The bacteria is died. Right? After the death of the bacteria, see, within the solution, the DNA is freely floating or freely present. This is the naked DNA. That naked DNA, for suppose any new bacteria is entered into that solution. So this DNA is also entered into this naked DNA molecules also enter into the bacteria. The naked DNA molecules are entered into another bacteria. So one particular bacteria DNA enter into another bacteria. So this is nothing but transformation. Entry of naked DNA molecules or foreign DNA molecules into another bacteria. Okay, the step is called as transformation. See, actually the name transformation means changing from one form to the another form. I will explain again in a very simple concept. This is a bacteria. This is pathogenic bacteria. Means have the capacity to cause the disease. And this is the bacteria. Non-pathogenic bacteria. See, as per my statement here, the pathogenic bacteria is dead, but the DNA is stable. The DNA is entered into the pathogenic bacteria DNA entered into the non-pathogenic bacteria. Then non-pathogenic bacteria modified into pathogenic bacteria. Because whatever that disease-causing genes are there, the disease-causing genes are entered into the non-pathogenic bacteria. Then the non-pathogenic bacteria become pathogenic bacteria. So a bacteria is modifying from non-pathogenic form to pathogenic form. So this step is called as transformation. Changing from one form to the another form. Right? So that is about the transformation. Right? So we know about one of the Griffith is the best scientist. We will discuss second year. We have the experiments also in this one. We will discuss. So in a very simple format, this is the transformation. Right? Naked DNA molecules or foreign DNA molecules are entered into another bacteria. Then changes the characteristic features of the bacteria. Right? That is about the transformation. And next is the conjugation. See, before going to discuss about the conjugation process, this is one of the bacteria. The bacteria having main genetic material and the bacteria having plasmidium. See, we know about the plasmids. Plasmids are extra chromosomal, circular, double-stranded DNA molecules. So those the bacteria which contains the plasmids, these cells are called as F plus cells. And some of the bacteria are there. So the bacteria are having main genetic material. And these bacteria are not having plasmids. So these cells are called as F minus cells. Plasmids are present, F plus cells. Plasmids are not present, F minus cells. And these cells are called as donor cells. And these cells are called as recipient cells. So these are recipient, these are donors. So why these are donors? Because see, whatever the plasmid DNA is there, the plasmid DNA transferred from F plus cell to F minus cell. That's the reason why this cell is called as F plus cell. Okay, this one is changing. That's the reason why this one is called as F minus cell. So how the transfer is taking place? We already discussed. See, this is one of the F plasmid responsible for the formation of the filament. See, this is the bacteria and this is another bacteria. What whatever that F plasmids are there, the F plasmids are produces the filling proteins. The proteins are called as filling proteins. The filling proteins are forms the villi. That is nothing but conjugation tube. See, this is the F plus cell, this is the F minus cell. See, presence of the plasmid, some of the proteins are synthesized. The proteins are produces one of the tube-like structure. These are nothing but sex villi. These are called as sex villi. Villi are two types, right? These sex villi are involved in the conjugation process. Sex villi are involved in the conjugation process, and these are also called, the sex villi is also called conjugation tube. These are also called conjugation tube. So F plus cell, F minus cell, and the sex villi, there is a conjugation tube. This is nothing but conjugation tube. Then whatever the F plasmids are there, the F plasmids are replicated. Replication is nothing but produces the same copy. Then the linear form. In the linear form, the plasmid DNA is transferred from F plus cell to F minus cell. In the linear form, the plasmid DNA is transferred from F plus cell to F minus cell. So this is the double-stranded DNA in the linear form, right? Then the DNA is transferred. Once the DNA is transferred from F plus cell to F minus cell, then this one is also, the F minus cell is also become F plus cell. The F minus cell also become F plus cell. So the process is called as conjugation. The process is called as conjugation. However, remember, within the conjugation process, whatever that F minus cells are there, the F minus cells are become F plus cells by the acceptance of the plasmid DNA from F plus cell. See, the plasmid DNA is transferred through the conjugation tube. This one is also called sex villi. So, through the conjugation tube, the plasmid DNA is transferred. That's the reason why this process is called as conjugation process. This process is called as conjugation process, right? That is about the conjugation. And the last one is the transfection process. See, this is a quite different one. See, here there is an intermediate agents are there. Here this is one of the bacteria, right? Bacteria having genetic material. Bacteria having genetic material. See, some of the bacteria attacked by the viruses. See, this is the one of the virus. The virus attacks the bacteria. The virus attacks the bacteria. Remember, these viruses are called as bacteriophages. These are called as bacteriophages. So you remember, whatever the viruses are there, some of the viruses which are attacks on bacteria, that viruses are called as bacteriophages. Okay, within the bacteriophages, whatever the genetic material is there, the genetic material is transferred into the bacteria. The genetic material, I'll explain step by step. This is the bacterial genetic material and this is the viral genetic material. The viral genetic material is transferred from here to here. That is nothing but from virus to cytoplasm of the bacteria. So how the transference takes place? Remember, the transfer here done by the injection of production of lysozyme enzyme. One of the lysozyme enzyme is there. The lysozyme enzyme involved in the cleavage of bacteria cell. Lysozyme is the enzyme. Lysozyme is the enzyme. That enzyme involved in the cleavage of bacteria cell wall. Once the bacteria cell wall is cleaved, then the viral genetic material is transferred from virus to bacteria cytoplasm. Finally, the viral genetic material injected into the bacterial genetic material. I already said viruses are intracellular obligatory parasites. Viruses are not having any machinery. Viruses are completely depends on host cells. So once the genetic material is inserted within the back to genetic material, then what is the use? Remember, once the viral genetic material is inserted, in that case, there is a new viruses are produced. New viruses are produced like this. This is the new virus, this is the new virus, and this is the new virus. Here, new viruses are produced with the genetic material. With the genetic material. So, here advantage is viral replication is takes place. Here advantage is viral replication is takes place. So, what is the advantage to the bacteria? What is the advantage to the bacteria? Remember, this is the viral genetic material. This is the viral genetic material. See, this is the viral genetic material. Along with the viral genetic material, the back to genetic material, a bit of back to genetic material is also transferred from one bacteria to the another bacteria. Remember, this is the virus, attacks the bacteria. The viral genetic material attacks with the bacteria genetic material, right? See, along with the, after that, again new viruses are produced. Within the new viruses, viral genetic material is present. Along with the viral genetic material, bacterial genetic material is also transferred. Again, these are attack on new virus. Again, these are attack on new bacteria. Again, this DNA shifted to the, or this DNA, okay, the bacterial DNA attached to the another bacteria. See, what are the ends? Bacterial DNA is present, the bacterial DNA attached to the another bacteria. Right? So, what I am saying here, don't think about here viral replication. Just think about here the transfer of bacterial genes from one bacteria to another bacteria by the help of the virus. So, this process is called as transfection. So, in all these cases, the common point is DNA is transferred. And the next common point is there is no new cell. Okay, there is no new cells production. So that's the reason why this is called as pseudosexual reproduction. Right? In the next class, we will discuss about the mycoplasma and the actinomycetes. Then we completed regarding to the Monera.